Hello and welcome to this episode of ESDR Kitchen. As you know by now, probably ESDR Kitchen is a webinar series that the ESDR organizes, started in the beginning of the pandemic and has been going on ever since. So today's episode is a part of the series called Freshly Baked, in which the latest high impact research findings are presented, really freshly baked papers. My name is Enikö, and I'm a senior lecturer at Uppsala University and at Karolinska Institute, Stockholm, Sweden. And I will be co-chairing this session with Professor Curdin Conrad in Switzerland. So we have two excellent speakers for today, Sebastian Willenborg and Jeremy Di Domizio, who will present really fresh, high impact, interesting papers. So I would like to start with presenting our first speaker, Sebastian Willenborg, who is a postdoctoral researcher at the University of Cologne at the Department of Dermatology in Germany. Just a few words about him. So he did his master thesis at the University of Marburg in Marburg, Germany, and then he was a PhD student in the group of Professor Sabine Emming in Cologne, Germany. And it seems like that he either fell in love with Cologne or with skin research because he has been there ever since. So I'm very much looking forward to your presentation. And for you who are listening, please don't forget, you can always ask questions and you are very much encouraged to do that in the chat or in the Q&A. And all questions for both presentations will be taken after the two talks. Please, Sebastian, the stage is yours.
Thank you very much for the kind introduction. Also, thank you for the opportunity to present our paper in the ESDR Kitchen. Yeah, so tissue repair is a highly complex process. And you can see here on the photographs that the outcome of a healing response in the skin can be very different, ranging from perinatal regeneration towards scar formation and also to pathological healing. And one major hallmark of chronic non-healing wounds is actually an altered tissue vascularization. So we ask in our group the question, how does inflammation and how do in particular macrophages regulate the outcome of the healing response? So what is the functional role of macrophages in physiological tissue repair? In our group, we use a mouse model of skin wound healing in which we perform full thickness punch biopsies in the back skin of mice. And you can see here that the healing response proceeds in sequential and overlapping phases of healing. Now, macrophages are key players in the regulation of the progression through these different phases. They are highly abundant at all stages of repair, and they exert phase-specific functions, as for example, they stimulate tissue growth and angiogenesis in the early and mid stage of healing. So we asked, how are these different functions regulated? And to address this question, we performed a bulk transcriptome analysis. Here we FACS sorted wound macrophages from black 6 wild-type mice. And you can see here in this volcano plot that we found clearly different activation profiles of macrophages. So in the early phase, at day 4 post-injury, we found a pro-inflammatory and pro-angiogenic profile, while in the late phase, we found a type 2 cytokine-driven pro-fibrotic profile. We then asked, what is the mechanistic regulation of these activation profiles? And one major finding in the last 5 to 10 years was actually that metabolic pathways and metabolites regulate the activation of immune cells. And here this cover in cell metabolism actually illustrates and highlights the complex balance between metabolism and immunity. So is this also true in tissue repair? Yes, we found that actually very important metabolic pathways, such as the glycolytic pathway, are highly upregulated in early stage wound macrophages, while in the late stage of healing, the cells induce genes involved in fat uptake, lipolysis, and fatty acid oxidation. And these transcriptional differences are important because we also could show in a functional assay in extracellular flux analysis that indeed the rate of glycolysis is higher in the early stage wound macrophages, while the late ones perform oxidative phosphorylation in the mitochondria. We focused then our study on the metabolic pathways and came up with the hypothesis that the metabolic shift from glycolysis towards OXPHOS in myeloid cells is critical for the repair response. And first, we wanted to impair OXPHOS, specifically in myeloid cells. And for this purpose, we made a new mouse model in which we bred the LysM-Cree mouse line to the DARS2 Flox mouse line. What is DARS2? It is an enzyme, a mitochondrial aspartyl tRNA synthetase. And you can see here in this cartoon that the gene is expressed in the nucleus. However, the protein is only active in the mitochondria. And here it catalyzes the specific attachment of aspartic acid to mitochondrial tRNAs. And this step is then essential for the synthesis of subunits of the electron transport chain. And this model works very nicely. So here you can see that when we culture bone marrow-derived macrophages, that in the absence of DARS2 protein, we have a severely disrupted electron transport chain. You can see this here for complex 1 and complex 4, which are virtually absent in the knockout cells compared to the controls. And also functionally, we could show that the oxygen consumption rate as measured by seahorse was significantly lower in the DARS2 deficient macrophages compared to the controls. So what is now the wound healing phenotype in these mice which have clearly an OXPHOS deficiency in macrophages? We found that the formation of vascularized granulation tissue was severely impaired in the knockout mice compared to the controls. So at day 7 in controls, we have a closed wound. We have also a nicely formed granulation tissue, which has also a high and dense network of blood vessels, as shown here. However, in the knockouts, we found beside a delayed wound closure, also impaired formation of granulation tissue. You see here also the fibrin still present in the wound tissue. And strikingly, also the angiogenic response was significantly attenuated in the knockout mice compared to the controls. And we found this very interesting because in this stage, we target actually the glycolytic macrophages. So what is now the link between impaired angiogenesis and the electron transport chain in the glycolytic early stage wound macrophages? To address this question, we checked first the levels of VEGF-A because this growth factor is in our model very important to stimulate angiogenesis. And you can see here the expression of VEGF is highly induced in control macrophages, and the expression is indeed attenuated in the knockout cells at day 4 post-injury. Now, interestingly, the VEGF is a HIF-1 alpha target gene. And we also found other HIF-1 alpha target genes being lower expressed in the knockout cells compared to the controls. So we asked, is the stabilization of HIF-1 alpha impaired in DARS2 deficient cells? The answer was yes. So when we cultured, again, bone marrow-derived macrophages, we could show that LPS and succinate induces in control cells the stabilization of HIF-1 alpha as shown here by Western blot. However, this response was completely absent in the DARS2 deficient cells. So what is now the link between HIF-1 alpha stabilization and the electron transport chain? The answer is it is the mitochondrial ROS. The mito-ROS are highly produced in inflammatory LPS activated macrophages. And you can see here in the scheme that the production of Mito-ROS was significantly attenuated in DARS2 deficient macrophages compared to the controls. So obviously a functional electron transport chain is required for the mitochondrial ROS production and subsequently for HIF-1 alpha stabilization in macrophages. And to summarize our major finding in our paper, I would like to point out that the induction of glycolysis in early stage wound macrophages is not sufficient to drive the pro-inflammatory and pro-angiogenic activation phenotype. They also need the repurposing of mitochondria from ATP production towards mitochondrial ROS production, which then activates HIF-1 alpha, and this important transcription factor can then induce VEGF and IL-1 beta, and we could show that VEGF is really the crucial mediator for the induction of angiogenesis. And if we now disrupt the electron transport chain, as we did in DARS2 deficient cells, we have lower levels of Mito-ROS, HIF-1 alpha cannot be stabilized, and we have less expression of its target genes, resulting finally in our tissue in impaired angiogenesis and also delayed wound healing. And we find this interesting because I think this is a good example of how a metabolic pathway regulates macrophage activation in an in vivo model of tissue repair. So what can we do with that finding? We ask then the question, how can we identify and target macrophage populations that repurpose mitochondria? And I can tell you already that by single cell sequencing, we could already find a subpopulation of macrophages which is likely to repurpose mitochondria. And we will characterize this in the future. The second question would be, is the mitochondrial ROS, HIF-1 alpha and VEGF axis dysregulated in human wound healing disorders, for example, in non-healing chronic wounds? And the last question is about the function of OXPHOS in late-stage wound macrophages. This question is still open for us, and we are working on that. Thank you very much for your attention. Also, many thanks to Sabine, who made the project possible, and also to our entire group, and also many thanks to our national and international collaboration partners. Thank you very much.
Well, thank you, Sebastian, for this excellent talk. Get to the questions. I'm sure there are several in the end. Again, please fill in your questions you have in discussion. We'll put them to the table for Jeremy and Sebastian then at the end after Jeremy's talk. And with that, it's my pleasure to introduce Jeremy Di Domizio. Jeremy, actually the second time he's presenting at the ESDR kitchen. That speaks for himself and for his track record and publications. Jeremy is currently a senior research scientist here in Lausanne, Switzerland, and is the supervisor of the dermatology research laboratories in Vigergier's group. He used to do his PhD in Grenoble, France, and then moved for a postdoc to Houston, where I actually had the pleasure to meet Jeremy already the first time, and then he moved here to Lausanne, so we could continue collaborating. Jeremy has an unbelievable track record on his publication list. He has several publications in papers like Nature Communications, Nature Immunology, Science, and others, and now since this year also in Nature. And he's going to present this paper on stinging interferon in COVID. So Jeremy, thanks for presenting again here.
Yeah, thank you, Curdin, for this nice introduction. And I will give you a brief summary of our recent findings on COVID-19. So as you may know, COVID-19 is induced by SARS-CoV-2. It's an RNA virus that infects the upper respiratory tract. And in 85% of cases, this leads to mild symptoms. But in 15% of cases, at least in non-immunized patients, this leads to severe disease with pneumonia and acute respiratory distress syndrome. And beyond the lungs, this inflammatory response and or the virus, can spread systematically to other organs affecting the brain, the heart, the kidney, and the skin. So this differential outcome in the disease is thought to be due to a difference in type 1 infection. So SARS-CoV-2 infect ACE2 expressing epithelial cells, and the RNA of the virus is detected either in endosomes by TLR3 or TLR7, or in the cytosol by RIG-I or MDF5, leading to the translocation of IRF and the production of type 1 interferon. But the kinetics of this type 1 interferon expression will dictate the disease outcome. So we observe that in mild disease patients, there is an early and robust production of interferon that can control the virus, whereas in severe disease, there is either a delay in this interferon response or a complete absence of the interferon response. And in the latter case, this was shown to be due to genetic mutation in the interferon pathways or the presence of neutralizing antibodies to interferon. So in this study, what we wanted to know is what drives the delayed interferon response in severe COVID-19. So what we did is to start analyzing skin lesions that were associated to severe COVID-19. We selected 3 purpuro-necrotic skin lesions and 7 maculopapular skin rash. We first wanted to know what kind of inflammatory response was present in this skin lesion. So for this, we performed transcriptomic analysis of the inflammatory genes by nanostring technology of those lesions. And we compared with the genes expressed by other inflammatory skin disease, including plaque psoriasis that express a Th17 signature, atopic dermatitis that express a Th2 signature, lichen planus that express a Th1 signature, and cutaneous lupus that express a type 1 interferon signature. And as you can see, we found that COVID-19 skin lesions clusterize close to lupus as they share this type 1 interferon signature. And comparing the two, we found that COVID-19 also expressed specifically a macrophage signature. So those signatures were also reported for the lungs, indicating there is a shared immunopathogenesis between skin and lungs. We first confirmed that this macrophage signature is actually due to an increased infiltration of macrophages in COVID lesion compared to lupus lesion. Then we wanted to know what are the cells that produce type 1 interferon, and we found that CD63 positive macrophages are the main producers of type 1 interferon in most of the patients we analyzed, followed by CD31 positive endothelial cells. And looking further at the cells, we found that macrophages tend to accumulate around blood vessels. And within those blood vessels, we found that endothelial cells were dying with vacuolization, here seen by electron microscopy, and the presence of nuclear Cliff-Caspar-3 fragments. So we also found that these dying Cliff-Caspar-3 positive endothelial cells were taken up by perivascular macrophages in some instance, which led to the transfer of DNA to the cytosol of macrophages. And more importantly, the macrophages that contain those cytosolic DNA were the one producing type 1 interferon, here seen by RNA staining. So what is the link between cytosolic DNA and the production of type 1 interferon? This is likely due to the activation of a cytosolic DNA sensor. And we thought about the cGAS-STING pathway, because cGAS is able to detect cytosolic DNA, producing a second messenger called cGAMP, that is the direct activator of STING, which leads to the dimerization and phosphorylation of STING and subsequent production of type 1 interferon and pro-inflammatory cytokines. So to assess whether this pathway was activated in this skin lesion, we first measure the quantity of cGAMP produced, and we found that indeed cGAMP was produced in the COVID skin lesion. And we also assessed the presence of phospho-STING in this lesion and found that perivascular macrophages, but as well as endothelial cells, were positive for phospho-STING. So what about the lung? For the lung, we used lung autopsies from COVID-19 disease patient. And we confirmed the presence of a strong type 24 response in those lungs, here seen by the MXS staining. And we confirmed the activation of the cGAS-STING pathway with presence of phospho-STING in both macrophages and endothelial cells. So in this first part, we see that in blood vessels, in skin lesion associated to COVID-19, endothelial cells have STING activated, they produce some type 1 interferon, but these endothelial cells also dying, taken up by perivascular macrophages, which leads to STING activation and production of type 1 interferon. The next question we ask is, what does activate STING in endothelial cells? So to answer this question, we use the lung-on-chip model, in which we have two independent channels separated by a membrane, in which we can culture lung epithelial cells on the airflow and lung endothelial cells on the vascular side, mimicking the alveolar capillary interface. And then we can infect the epithelial cells with the SARS-CoV-2 and perfuse a STING inhibitor, the H151, in the vascular side. Doing so, we found that upon infection, there is induction of interferon beta production in the adjacent endothelial cells. Also, there is induction of cell death of those endothelial cells, and both interferon and cell death was blocked by the STING inhibitor. So to confirm the role of STING in these settings, we cultured different cell types, white type or STING knockout cells, epithelial and endothelial cells. We found that STING knockout in epithelial cells didn't affect the cell viability, but STING knockout in the endothelial cells could prevent the cell death induced by infection. So we looked further at those endothelial cells, and we found by electron microscopy analysis that upon infection, the mitochondria of those endothelial cells were swollen and they were losing their cristae formation. And this loss of cristae formation was also found in mitochondria in endothelial cells from the COVID skin lesion, indicating that there is probably a release of mitochondrial DNA that may trigger cGAS-STING. So to confirm this hypothesis, we treated the lung-on-chip with either the 2', 3' DDC that deplete mitochondrial DNA, or with VBET4, which inhibits the release of mitochondrial DNA. In the two cases, we could inhibit the induction of interferon beta in endothelial cells. So finally, to confirm that this pathway is important in the COVID disease, we used an in vivo model using transgenic mice transgenic for human ACE2 that we infected with SARS-CoV-2 and treated with the H151 STING inhibitor, either in a prophylactic settings, so starting the treatment before infection, or in a therapeutic settings by starting the treatment 2 days after infection. And in both treatments, we could reduce lung inflammation in the infected mice, as well as the type 1 interferon response. But more importantly, we could reduce the weight loss in the infected mice and improve the survival of those mice. And of note, the STING inhibition did not affect the viral load in the lungs. So altogether, we can say that following infection of the lung epithelial cells by SARS-CoV-2, this leads to mitochondrial disruption in the adjacent endothelial cells, leading to the release of mitochondrial DNA that activates cGAS-STING. This leads to type 1 interferon production, but also to the cell death of those endothelial cells that are taken up by perivascular macrophages. This activates cGAS-STING in macrophages and the production of type 1 interferon and pro-inflammatory cytokines, triggering the cytokine storm observed in severe COVID. So we think that inhibiting STING with STING inhibitors is a promising therapeutic strategy to treat severe COVID-19. So with this, I would like to thank all the people who participated to this work. It was a close collaboration between two groups. So the group of Michel Juillet in the University Hospital of Lausanne, and we would like to thank in particular Fanny Sedoun and Ahmad team who performed the analysis on human samples, and the group of Professor Andrea Blasser at EPFL. And I would like to thank Fatih Gulen and Vivek Thakkar who performed the lung-on-a-chip models and the in vivo models. And I'm finished here, and I will be happy to answer your question.
Thank you very much, Jeremy. Excellent talk. First questions are coming in. So I'll start with the very first that came in for Sebastian, if he's also activating his camera. So first of all, also a very nice project from Pia Saha. And there's a question about regarding the MKO and the question if every macrophage is or every macrophage has the KO for DARS2, and if there would be more specific, if it would be for Langerhans cells.
Yes, a good question. So we use the lysozyme M3 mouse line, which targets in our wound healing model between 70 and 80% of the macrophages in the wound tissue. So a mouse line which targets only the Langerhans cells would definitely be more specific than our LysM3 mouse line. Yeah. I think our LysM3 mouse line also targets the Langerhans cells in the newly formed epidermis in the wound tissue. Yes.
Thank you. There's also
go ahead, Enikö.
Yeah, thank you. There's also a question to you, Sebastian, from Ritu Bansal, who's thanking for the nice presentation as well, and asks about the, in the early stage, you said that according to your initial findings, glycolysis is more important, but how then does the DARS2, which impacts the oxidative phosphorylation, impact wound healing in the angiogenesis phase? And also, what do you mean by repurposing the mitochondria?
Yeah, a good question. And that's exactly the surprising finding that we had. So initially, when we started the project, we aimed to blocking the progression from the inflammatory towards the resolution macrophage in the late phase of healing, which depends on OXPHOS. However, to our surprise, we found already a severe wound healing defect in the early and mid phase of healing in the angiogenic response, for example. And our key finding in the paper was that the early stage wound macrophage does not use the mitochondria for ATP production, so not for OXPHOS, but it needs the mitochondria for ROS production. And only then it can drive inflammation and angiogenesis. So it is the glycolytic induction, but also the ROS production in the mitochondria. And repurposing means that it does not use the mitochondria for the ATP production, but ROS production.
Maybe a personal follow-up question on that one. Do you think there could be the same system which is not properly working in chronic wounds which don't heal, which become like atonic, they don't heal, they're not inflamed anymore, and anything that you have a failure in that system to actually activate this angiogenic part?
Exactly. So we would guess that in a condition of non-healing chronic wounds, this axis is disturbed, that somehow the macrophages cannot produce a sufficient amount of mitochondrial ROS, or maybe too much even, we don't know. So I think the level of mito-ROS is here important. No.
So, yeah, go ahead, Enikö.
Yeah, there is another question, which actually very much something that I have been thinking about when you presented this project. So Jakob Wikström is asking, how would you envision a clinical therapy that selectively targets the metabolism of macrophages? And this is exactly what I have been thinking when you're talking, how can you really selectively target one cell type or maybe one cell subpopulation?
Yes, that's a very good question, and also for us a challenge. But I think a good starting point is the single cell sequencing that we have already and that we have also published in the paper. And there we found actually 7 clusters of macrophages, and one of them showed characteristics of mitochondrial repurposing. So it was low in OXPHOS, it was highly angiogenic, it was ROS producing, and had and had also a broken Krebs cycle. So all hallmarks of mitochondrial repurposing. And I think we need to find now surface markers which characterize this subpopulation, and then one could at least start in mouse models to deplete this population specifically with Cre lines. Yeah. That would be my starting point.
Thank you.
Thanks. So to Jeremy from Sabine Hemming, thanking you also for your great presentation. Could you test your findings in a KO mouse model, which would be specific for or myeloid cell specific? I'll just give you all the three questions because it's like following up on one and after other. Is this process specific for perivascular macrophages? And how could you, if so, target this subset of macrophages?
So actually, so we have never tried to do STING, to use STING knockouts specific to myeloid cells for the mice, but of course it's one of our questions. Is it the STING pathway in endothelial cells or in macrophages the more pathogenic in these settings? This we don't know, so that would be interesting actually to test endothelial cells on one side versus myeloid cells on the other side to see what is more pathogenic for the SARS-CoV-2 infection. And then what was the second part of the question? How to target? Well, actually, for the therapeutic strategy, we don't think about targeting. It would be an oral treatment of STING inhibitors, yeah, like we did in the mouse. I'm not planning to have a more specific target, actually.
Yeah, there is a question also from Leopold Eckhardt. Thanks, excellent. And if these effects are specific for SARS-CoV-2 or do other viruses cause similar responses? Maybe I could add an own question also. Is this pathway also relevant for other inflammatory disease, autoinflammation?
So it could be. So actually, we showed that with the SARS-CoV-2, but there's probably other pathologies, so either virus-infected lungs or bacteria-infected lungs, or even other kind of inflammation of the lungs. Is there, if there is a damage of mitochondria in endothelial cells at some point, I don't see why it wouldn't be the same mechanisms with perivascular macrophages taking up those dying cells and having the STING activating. So this is something to investigate. Yeah.
Maybe following up on that a bit on the virus specificity. Pierre again asking the cGAS-STING is more specific for double-stranded DNA viruses. So how does mRNA virus like SARS-CoV-2 come into play?
So actually, maybe I was very fast, so it was difficult to have all this information in 10 minutes. But what we showed, the STING pathway is not directly activated by the virus. So the virus is RNA virus, and it leads to some type interferon by other sensors. And this actually is a good interferon response, a beneficial interferon response. It's early and it's seen in mild disease patients. But when the STING pathway is activated, this is due to the mitochondrial DNA released by damaged mitochondria or by DNA taken up by macrophages. So it's a side effect. So it's not direct effect of the virus. It's really a side effect of the infection.
Maybe just the last questions for you before we go back to Sebastian. Are there any similarities in cell death mechanism between COVID-19 and skin lesions like 10? And maybe I just add similarities maybe with the COVID-TOS, because it's also a late reply or response.
Yeah, I've really no clue whether it's similar with the 10. I would say in the transcriptomic analysis, this seems different because I don't see a macrophage signature in 10. So at least on the cell types are different. Yeah, then I don't know at the endothelial cell level, I don't know. Cannot say.
it's usually less inflamed than the COVID lesions that we have. Enikö, do you want to have a last question to Sebastian?
I think we have one more question, but I think that this is already answered.
Okay.
Yeah, with the macrophage phenotypes, I think with the single cell sequencing, that's already answered.
Right, so you're good. So with that, I first like to thank the two speakers for really two excellent, outstanding, not just presentation, but also publications, and congratulations to the entire groups. With that, I'd like to just give you some information. The next kitchen will be on sweet and sour in actually not in 2 weeks, but in 4 weeks. So it's April 20th. Also for the submission of the ESDR abstracts, not just to register for the ESDR in late September, not October, September 28th. So the deadline for the submission is May 27th, in order to actually then celebrate the first again face-to-face meeting in Amsterdam and in late September. So hoping to see you all there and have a really good time again after this COVID and now horrible times in Ukraine. Just hoping everything is passing fast in the name also of the ESDR. You know, we've gave some money to the Red Cross, but we support anyway all support of the people there, and that this situation is resolved as quickly as possible, hopefully. So with that, I'd like to thank you all for attending, and all the best. Stay safe. Thanks a lot.
Thank you.
Thank you very much.